Hello friends, now we will start part 2 of the lecture on the summary of the book Brain Energy written by Dr. Christopher M. Palmer. Dr. Palmer provides an insight into the origin of mental disorders and the methods of treating them. Dr. Palmer's theory can usher in potent therapeutic interventions for pervasive psychiatric conditions for which standard pharmaceutical approaches have proved to be minimally effective. Dr. Palmer says that mental disorders are due to an energy imbalance in the brain, and mental disorders have a metabolic connection to them. Mental disorders appear to be linked to metabolic disorders like diabetes because direct evidence of metabolic abnormalities have been found in people with mental disorders. And these abnormalities have been found in the markers of metabolism. Now, what are these markers of metabolism? Differences in levels of ATP, redox markers, that is balance between oxidants like reactive oxygen species and antioxidants, differences in levels of hormones, differences in neurotransmitters, lactate, which is a marker of metabolic stress, cortisol dysregulation appear to play a role in at least some people. So these biomarkers, differences in these biomarkers have been found in people with mental disorders, and these are metabolic indicators. In 2017, Dr. Wallace, founder of the field of mitochondrial genetics, published an article claiming that all psychiatric disorders are a result of mitochondrial dysfunction. So mitochondria are the common pathway for all mental as well as metabolic disorders. And psychiatric disorders, that is disorders of the mind, they are a result of dysfunctioning of the mitochondria. Dr. Wallace focused on the mitochondrial genes. Mitochondrial genes, they mutate due to reactive oxygen species. We studied in the part 1 of the video that mitochondria are involved in producing and cleaning up of reactive oxygen species. And mitochondria also get affected by these reactive oxygen species because mitochondrial DNA are often unprotected, and they get influenced when the reactive oxygen species are large in number. When large number of reactive oxygen species accumulate, they become toxic and they adversely affect the mitochondria. And mitochondrial genes may mutate due to reactive oxygen species. We studied in part 1 of the video that the reactive oxygen species, how are they produced? When food like carbohydrates, proteins, fats, etc. is burned to produce energy, then electrons, they flow along the electron transport chain. So this energy is in the form of electrons flowing through the electron transport chain, and some of these electrons, they leak out of the system. When they leak out of the system, they form the reactive oxygen species like H2O2 or superoxide ion, etc. And these reactive oxygen species, they have some beneficial effects also. They serve as important signaling molecules also, but when they accumulate in large numbers, then they become toxic. And since mitochondria are involved in producing these reactive oxygen species and cleaning up these reactive oxygen species, when they accumulate in large numbers, they prove to be detrimental to mitochondria also, because mitochondrial DNA is unprotected, and the reactive oxygen species, they can cause mutations in the mitochondrial DNA. Now, Dr. Wallace argued that brain is the organ that is most affected by the problem of energy production by mitochondria, because we know that it is always the most vulnerable part which will be affected. And since brain is the most sensitive and vulnerable organ, so it is most affected by the problem of energy production by mitochondria. So if mitochondria are producing less amounts of energy, then the brain will be most affected. We studied in the part 1 of the video that the mitochondria make up 40% of the brain cells. So they are even more important for the brain than they are for the body. Dr. Wallace argued that different parts of the brain might fail first, likely because they are more sensitive to energy deprivation than others. So the brain has got different parts. Some parts will be more sensitive to energy deprivation, while other parts may be relatively less sensitive to energy deprivation. So the parts of the brain which are more sensitive to energy deprivation, they will be failing first. Now some of the scientists and researchers, they are rebutting this theory given by Dr. Wallace, and they say that focusing solely on energy production by mitochondria cannot account for the diversity of symptoms that we see in billions of people with mental disorders. So if we focus solely on the energy production by mitochondria, then we cannot account for the diversity of symptoms that we see in billions of people with mental disorders. So people with mental disorders, they are displaying diverse number of symptoms, and if we focus solely on the energy production by mitochondria, then this cannot explain all the symptoms that are experienced by the people with mental disorders. Another point in rebuttal is that people with the same mitochondrial genetic mutation, so people who are having the same genetic mutations in the mitochondria, they can have different symptoms. So two persons may be having the same genetic mutation in the mitochondrial DNA, and but they can still display different symptoms. So these are 2 points of rebuttal given by certain researchers. But now these researchers, they failed to discuss, they failed to consider how many other roles mitochondria play in the cells apart from energy production. So they did not consider that mitochondria are not merely energy producers. They are playing a lot of many other important roles as well. Like they are fusing with each other. They are fusing with each other. They are sending signals to other cells. They are playing a role in the production of neurotransmitters and hormones. They are cleaning up reactive oxygen species. They are having myriad number of roles. And these researchers who are rebutting the theory given by Dr. Wallace, they did not consider the other roles played by mitochondria. Now, mitochondria, if certain mitochondria are not producing enough energy, then they may also not be able to fuse with each other. And if they are not able to fuse with each other, then it may adversely impact other functions performed by mitochondria also. And they may not be able to send signals to the cells. They may not be able to trigger the various epigenetic mechanisms in the body, that is turning off genes on and off. So a large number of other functions of mitochondria may also be affected, and focusing solely, so it is not merely energy production by mitochondria, there are large number of other roles that are also being played by the mitochondria. And the researchers who are rebutting the theory given by Dr. Wallace, they fail to recognize how many different factors affect the function and health of mitochondria apart from genetic mutation. So they are just focusing on genetic mutation. It is not only genetic mutation which is affecting the functioning of mitochondria. There are large number of other environmental factors also, like hormones, neurotransmitters, and other factors which are affecting the health of mitochondria, which are affecting the functioning of mitochondria. So there are large number of factors to consider. This is what this theory of brain energy says. Now, Dr. Palmer says that there is a shortfall in the research on the role of mitochondria. What is this shortfall? One is that we are focusing only on one function of mitochondria, that is ATP production, that is energy production. So mitochondria, it is true that they are the powerhouse of the cell, but they are also performing large number of other functions, and we are not focusing on other functions. So mitochondria are not producing enough ATP. If the mitochondria, they are not producing enough ATP or energy, then they might have other defects also. They are not producing enough ATP, then they may have trouble fusing with each other two mitochondria may have trouble fusing with each other because of lack of energy production. Then they may be leaking large amount of reactive oxygen species. So leaking large amount of reactive oxygen species may again create problems, may create toxicity for the cell and for the mitochondria. And they might have trouble managing the calcium levels in the cell. We have studied in the part 1 of the video that mitochondria are involved in calcium regulation in the cell. And if certain mitochondria are not producing enough ATP, then they may also have trouble managing calcium levels in the cell. We know that there is a resting, neurons have got a resting membrane potential. The inside of the cell has got a negative charge compared to the outside of the cell, and this negative charge is created by ion pumps, which includes pumps which pump ions like sodium, potassium, calcium, etc. into the cell or outside the cell or within the cell. So these ion pumps are responsible for the resting membrane potential of neurons, and triggering of these ion pumps leads to the cell doing its task or performing its function. And mitochondria are involved in this process of ion pumping and in calcium regulation. So we are only focusing on one function of mitochondria, that is energy production. We need to focus on the other functions as well. And all these other functions like mitochondria fusing with each other, sending signals, leaking, mitochondria playing a role in the regulation of reactive oxygen species, managing calcium levels in the cell, these functions may not be clearly measurable. It may not be possible to measure these functions accurately. Then the next point is about the shortfall in research on the role of mitochondria, that some cells may have perfectly healthy mitochondria in abundance. So some cells might have perfectly healthy mitochondria in abundance while others may not have. So if you are studying a healthy cell, then it will provide you no clue regarding what is happening in a diseased cell, what is happening in another unhealthy cell. So we need to focus on specific cells, specific target cells. If you are studying cells which are perfectly healthy mitochondria, may provide no clue about what is the problem with the person, what is happening in a diseased cell. So studying a healthy immune cell may not offer any information about what is happening in a malfunctioning brain cell. Then the third shortfall in the research on mitochondria is the role of feedback loops. Now what are feedback loops? It means that everything is interconnected. So causes can lead to consequences, but consequences can also lead to causes. For example, mitochondria are regulating the reactive oxygen species. They play a role in the production and cleaning up of reactive oxygen species, but the reactive oxygen species also influence mitochondria. If large number of reactive oxygen species accumulate, then they can cause mitochondrial genetic mutations, they can damage mitochondria. Similarly, mitochondria are playing a role in the production and regulation of neurotransmitters, but neurotransmitters are also affecting mitochondria. Mitochondria have receptors for some neurotransmitters like serotonin. So mitochondria have receptors for serotonin, and serotonin also plays a role in increasing the ATP production in mitochondria. So there are feedback loops everywhere. There are interconnections everywhere, and current research is not focusing on these interconnections. Now we come to a neurodegenerative disorder, that is Alzheimer's disease. Now Alzheimer's disease is caused by the accumulation of a protein called beta amyloid in the brains of people with this disease. And this protein, beta amyloid, it is toxic to the mitochondria and it causes mitochondrial dysfunction. Now this occurs due to maintenance problems in the cells. The cells are unable to maintain themselves due to mitochondrial dysfunction. And again it is a feedback loop. There is mitochondrial dysfunction, mitochondria are not functioning properly. This may lead to debris accumulation in the cell, accumulation of beta amyloid in the cell. And this beta amyloid in turn is toxic to mitochondria. It causes further mitochondrial dysfunction. So this is again a feedback loop. Now, what causes this mitochondrial dysfunction? There is evidence that this mitochondrial dysfunction begins even before beta amyloid starts accumulating. So this mitochondrial dysfunction is starting first, and there may be a positive feedback loop. Mitochondrial dysfunction leading to maintenance problem in the cell, leading to accumulation of this beta amyloid, because this protein should be getting recycled and disposed of, but it is not getting disposed of. It is accumulating as debris in the cell, and this accumulation makes the mitochondrial dysfunction even worse. And this leads to a downward spiral which causes Alzheimer's disease. So maintenance problem in the cells is causing the accumulation of beta amyloid, which is causing Alzheimer's disease. Now, once this mitochondrial dysfunction starts, then it can lead to, because all the regions in the brain, they are interconnected. So if one part of brain is affected, it will affect other parts of the brain also. And this interconnection can lead to other symptoms of Alzheimer's also. Now, mitochondrial dysfunction, why is it caused? Either due to genetic mutations in the mitochondria, accumulation of too much of reactive oxygen species. Second cause of mitochondrial dysfunction is shortage of mitochondria in the cells. When there are inadequate number of mitochondria in the cells, when there is a shortage, then it causes mitochondrial dysfunction. If defective mitochondria are not disposed of and recycled, if the mitochondria that are defective, if they are not disposed of, if they are not recycled, then it can cause mitochondrial dysfunction. And aging. Now, mitochondrial dysfunction can be a cause of aging, because if number of mitochondria decrease, then the metabolic capacity of the cells will decrease. If number of mitochondria are decreasing, then the cell's metabolic capacity is decreasing, because enough energy is not being produced. Enough signals are not being sent to other cells. Enough hormones or neurotransmitters are not getting produced. So metabolic capacity of the cell decreases, and this causes aging. And we see accelerated aging in people with all mental disorders. Now, mitochondrial dysfunction means mitochondria are not functioning properly. And now, mitochondrial dysregulation. It means that mitochondria are not functioning properly due to environmental factors, extrinsic factors like neurotransmitters, hormones, peptides. Peptides, we know they are a chain of amino acids. They are a chain of amino acids. They are parts of protein. Then inflammatory signals and even alcohol. So environmental factors like drugs, alcohol, neurotransmitters, peptides, inflammatory signals, they impact mitochondria and they may cause mitochondrial dysregulation. So when environmental factors are playing a role, it is similar to people doing the best they can when they are highly stressed. So when we are highly stressed, sometimes the environmental factors are so overwhelming that we cannot do much. So in the same way, environmental factors are adversely affecting the functioning of mitochondria. So sometimes it is said that when a flower does not bloom, you should fix the environment in which it is present rather than the flower itself. So sometimes the problem can be with the environmental factors that are causing mitochondrial impairment. Then measuring levels of ATP to ADP. Now when we measure these ATP to ADP levels, they are an important biomarker. They tell us about the health of mitochondria in the cell. Now ATP levels, they are important signals within the cell. The ratio of ATP to ADP affects different aspects of cell function, including gene expression. So the ratio of ATP to ADP affects gene expression. That is, which genes are getting turned on and off. Now actually the genes in our brain, in our liver or in our heart, the DNA in our brain, the liver and heart is the same. But the difference is in the gene expression. So the different genes are getting expressed in the brain, liver and heart, because of which they are performing different functions. So genes, when they express themselves, they convert into proteins, and the specific cells start functioning. So the difference in function of these organs is caused not due to the difference in the DNA, but due to the difference in the gene expression. So the difference in functions is caused due to difference in the gene expression. Now, reduced levels of ATP have been found in mental disorders like schizophrenia, bipolar disorder, major depression, alcoholism, post-traumatic stress disorder, etc. So this indicates a mitochondrial dysfunction in people with mental disorders. Then other disorders like autism, OCD, Alzheimer's, epilepsy, cardiovascular disease, type 2 diabetes, obesity. In all these disorders, there is reduction in levels of ATP. Now we know that mitochondria are creating the reactive oxygen species and also help detoxify them through antioxidants. So mitochondria are involved in producing as well as cleaning up of the reactive oxygen species. When mitochondria are not working well, then reactive oxygen species will build up. This will cause damage to cell and damage to mitochondria. And this is a vicious feedback cycle. So mitochondrial dysfunction affecting increasing amount of reactive oxygen species and buildup of reactive oxygen species, further adding to mitochondrial dysfunction. Studies found higher levels of oxidative stress in essentially all metabolic, neurological, and mental disorders. So studies have found higher levels of oxidative stress, that is more number of reactive oxygen species in essentially all metabolic, neurological, as well as mental disorders, and causing cell damage as well as accelerated aging. So decrease in the metabolic capacity of the cell, mitochondrial dysfunction is increasing the oxidative stress, and this has been found in all metabolic, neurological, as well as mental disorders. Now, mitochondrial dysfunction can cause problems with cell maintenance that can lead to maintenance problems and structural defects found in people with mental disorders. So, in people with mental disorders, cells have maintenance problems as well as structural defects. In some cases, structural defects in cells can lead to a positive feedback cycle that affects metabolism. So structural defects in cells can lead to positive feedback cycle. So there is a structural defect in the cell. This is adversely affecting the metabolism, adversely affecting the mitochondrial function, and the mitochondrial dysfunction is further adding to the structural defect in the cell, further making it harder for the cells to work. For example, myelin. Now what is myelin? Myelin is the outer protective coating for neurons made by support cells called oligodendrocytes. Now, a neuron, a nerve cell, we know it has got a cell body called the cytosol. This is the cell body. These are dendrites that are coming out of the cell body. Then there is an axon, and this axon is covered by myelin sheath to protect it. Now, what is myelin doing? Myelin is protecting the neurons, and it is preventing the excess ions, etc. from entering the neurons. Only the adequate number should enter. So myelin is acting as a protective coating for the neurons. And myelin is made of support cells known as oligodendrocytes. These oligodendrocytes are support cells, and myelin is made up by these support cells called oligodendrocytes. Now myelin makes it easier for neurons to send electric signals. So this myelin coating, it is facilitating the sending of electric signals by neurons. It prevents the loss of message. If a neuron has deficits in its myelin coating, now suppose if this myelin coating is not there and there are defects or leakages in the myelin coating, then the neuron will require more energy to work. So if there are defects in this myelin coating, then the neuron will require more energy to work. And an extreme example of this is the disorder called multiple sclerosis. So what happens in multiple sclerosis? In this, the myelin coating of the neurons, it is destroyed by an autoimmune process. So when this myelin coating is destroyed, by the immune system attacking itself, an autoimmune process, then a disorder called multiple sclerosis takes place. Now, mitochondrial dysfunction has been associated with problems in myelin production as well as maintenance. Now see, when this myelin is not there, this myelin coating is not there, or there are defects in this myelin coating, then sometimes ions like sodium, potassium, calcium, etc. can leak into the cells. And these ions, we know that these ion pumping mechanisms are responsible for firing of the cell, which leads to the cell doing its task. Now when these ions are unnecessarily leaking into the cells, when they should not, then it can cause the cell to fire when it should not. So it can cause the cell to become hyperexcitable. And this hyperexcitability has been seen in symptom of psychosis, which is a symptom of schizophrenia or schizoaffective disorder. So problems in myelin coating, defective myelin coating can also cause psychosis. Now mitochondrial dysfunction has been associated with problems in myelin production and maintenance. Consistent with this brain energy theory, defects in myelin have been identified in brains of people with schizophrenia, major depression, bipolar disorder, epilepsy, in which there are seizures, then Alzheimer's disease, diabetes, and obesity. So defects in myelin have been identified in the brains of people with both mental disorders as well as metabolic disorders. Now, debris in the cell. Accumulation of debris in the cell. Now, this is a structural defect and a maintenance issue. Now, we just talked about Alzheimer's disease. Now, in Alzheimer's disease, we found that there was a maintenance issue in the cell and there was accumulation of beta amyloid. There is also an accumulation of a protein called tau in addition to this beta amyloid. Now, what are the effects of this tau protein? This protein severely limits the mitochondria's ability to move around the cell. So, this tau protein is blocking the movement of mitochondria. It is limiting the ability of mitochondria to move around the cell. It's obstructing the path of mitochondria. And tau protein interfered with the cytoskeleton that the mitochondria use for movement. So the mitochondria use a cytoskeleton for movement and tau protein is interfering with that. And when the mitochondria are not allowed to move around, then the cell will have trouble in working properly because the mitochondria, they move around to sites, to places where things are always happening. We studied in the part 1 of the video that mitochondria are involved in the production of neurotransmitters. So when neurotransmitters are produced, then they are packaged into vesicles, then they travel down the axon of the neuron to the sites called synapses, and then the neurotransmitter is released at the receptors of the target cells. So when mitochondria are prevented from moving around, this process of making neurotransmitters will be obstructed. So cell will have trouble working properly, and because of this, the cell may shrink or it may die. Now, how mitochondrial malfunction leads to mental illness. Now, human illness is usually due to a problem in one of the three areas: the development, function, or maintenance of cells. Either there is a problem in development of cells, which may result in neurodegenerative disorders, or there is a problem in function of cells. Cells may become overactive or cells may become underactive, or there is a problem in the maintenance of cells. Cells are unable to maintain themselves, causing accumulation of debris in the cell. And this we have just studied, it may lead to Alzheimer's disease. Now, 5 broad consequences of mitochondrial dysfunction and dysregulation. We know mitochondrial dysregulation means mitochondria are being adversely affected due to environmental factors which may be like alcohol, like drugs, or hormones, or neurotransmitters, neuropeptides, etc. So the 5 broad consequences are: first, reduced cell maintenance. So when cell maintenance is reduced, cell is not able to maintain itself. Then it causes debris accumulation in the cell. And debris accumulation can cause Alzheimer's disease, we have just studied. Now, overactive brain functions. When the cells of the brain, they become overactive. This can be because of mitochondrial dysfunction, hyperexcitability causing psychotic symptoms. Then underactive brain functions, when the brain cells have slowed down, they are underactive in performing their function. This can also be a consequence of mitochondrial dysfunction. Also developmental problems, like the brain neurons are not developing properly. This may lead to neurodegenerative disorders like Alzheimer's disease, autism, etc. Then cell shrinkage and death, when mitochondria are lesser in number of mitochondria are reduced. Some of them don't go to the peripheral parts of the cell. And this lack of energy production, when the peripheral parts of the brain are not they don't go to the peripheral part of the brain when they are less in number, because the workforce is not adequate. So when they don't go to peripheral parts of the brain, this may lead to death of cells in the peripheral part of the brain, leading to cell shrinkage and death. So this is also a consequence of mitochondrial dysfunction. So all these things are seen in mental disorders. Now we will study each of them in detail. First is reduced cell maintenance. So one study estimates that about 1/3 of our brain's ATP goes into cell maintenance. So 1/3 of the ATP in the brain is going into maintenance of the cell. Now how is cell maintenance taking place? Autophagy. Autophagy means the defective parts of the cell are getting recycled. The cell is repairing itself. There is a role of stress, cortisol and mitochondria in autophagy. So in the repairing of the cell parts, stress, cortisol, which is a stress hormone, and mitochondria, they play a role. Now mitochondria interact with other organelles of the cell to facilitate routine maintenance functions. For example, these mitochondria, they react with the lysosome for disposing of the waste products. Lysosome is the organelle of the cell which is involved in disposing of the waste products. And when mitochondria interact with these lysosomes, then lysosomes, they start performing their functions of disposing of the waste products. When interactions of mitochondria are with the lysosomes are prevented, then waste products will accumulate in the lysosomes. And when waste products are accumulating in large numbers and then they are not getting cleaned up, waste products will accumulate in the cell, and when these waste products are not getting cleaned up, they are not getting disposed of, then the cell may die. So this is a maintenance problem. Then mitochondria also interact with the endoplasmic reticulum, which is another important organelle in the cell, which has many roles, including the folding of proteins. So this endoplasmic reticulum in the cell is involved in the folding of proteins. And many neurodegenerative disorders are associated with misfolded proteins in the endoplasmic reticulum. So when the protein folding in the endoplasmic reticulum is not taking place properly, then a lot of neurodegenerative disorders may result. And when these misfolded proteins, they accumulate, a process called unfolded protein response tries to mitigate the damage. When this folding is not taking place properly, when these misfolded proteins accumulate in the endoplasmic reticulum, then a process called unfolded protein response takes place to mitigate this damage. Now researchers found that a microprotein in the outer membrane of the mitochondria, so there is a microprotein in the outer membrane of the mitochondria, and this microprotein, it plays a role in unfolded protein response. So this microprotein known as Big Boss, it plays a role in the unfolded protein response which tries to mitigate the damage caused by misfolded proteins in the endoplasmic reticulum. And when this protein Big Boss was eliminated, then the cells were much likely to die. So mitochondria are interacting with the endoplasmic reticulum for folding of proteins. And when mitochondria are not interacting with them, when these interactions are prevented, then it results in misfolded proteins in the endoplasmic reticulum. There is a microprotein in the... Now what happens is, I'll again repeat to make it clear. First is that mitochondria are interacting with the endoplasmic reticulum and they are playing because endoplasmic reticulum is playing a lot of roles, including the folding of proteins. So the endoplasmic reticulum is involved in the folding of proteins. And when misfolded proteins accumulate in the endoplasmic reticulum, then a process known as unfolded protein response tries to mitigate this damage, and in mitigating this damage, mitochondria are involved. How are they involved? Because in the outer membrane of mitochondria, there is a microprotein called PIGBOS. And this microprotein in the outer membrane of mitochondria plays a role in the unfolded protein response, which tries to mitigate the damage caused by misfolded proteins in the endoplasmic reticulum. So, in simple language, the mitochondrial proteins, they are preventing neurodegenerative disorders by ensuring that misfolded proteins do not accumulate in the endoplasmic reticulum. And when this mitochondrial protein, that is PIGBOS, when it is eliminated, then the cells are likely to die because the misfolded proteins will accumulate and will cause neurodegenerative disorders, will cause death of cells leading to neurodegenerative disorders. So mitochondria are preventing neurodegenerative disorders, and when there is a malfunctioning of mitochondria, then it can cause neurodegenerative disorders. Now we have just seen that maintenance problems in the cells can lead to mental disorders. Maintenance problems, first is mitochondria are interacting with the lysosomes for disposal of waste products. When this interaction is prevented, then waste products will accumulate, causing maintenance problems and resulting in mental disorders. Second, mitochondria are interacting with another organelle in the cell, endoplasmic reticulum, for ensuring proper folding of proteins. And when mitochondria are interacting with the endoplasmic reticulum, and endoplasmic reticulum is involved in the folding of proteins, and mitochondria are preventing neurodegenerative disorders. How are they preventing? The endoplasmic reticulum is involved in folding of proteins. When there is misfolding of proteins in the endoplasmic reticulum, then a microprotein which is there in the outer membrane of mitochondria called as PIGBOS. This tries to mitigate the damage by means of unfolded protein response. And when this microprotein was eliminated, then the cells were likely to die. So mitochondria are preventing neurodegenerative disorders by ensuring proper functioning of the endoplasmic reticulum. Now we come to overactive brain functions. This means that the brain cells are becoming hyperexcited. They are more often or more intense. The cells, they are working more often, or they are working more intensely than they should be, or at the wrong time. For example, seizures. Seizures is abnormal electric activity in the brain. Then heart arrhythmia, that is irregular heartbeat. Psychosis. Psychosis means that the brain cells are triggering when they should not. We know that during night we have dreams, and certain cells become active to produce dreamlike experiences. But these cells, they should ideally be shut off during the daytime. But when these cells are not getting shut off, when the brakes are not being applied, and even during daytime we are having dreamlike experiences, which we call as hallucinations. We are seeing things or hearing things which are not there. Then this means that our cells have become hyperexcitable, and they are causing symptoms of psychosis. Then muscle spasm. Muscle spasm is involuntary twitching of muscles. This is also an example of overactive brain cells, hyperexcitable cells in the brain or in the body. Then chronic pain. Hyperexcitable nerve cells are causing chronic pain. Then mitochondrial dysfunction can cause overactivity or hyperexcitability. When mitochondria are not functioning properly, then this may result in overactivity or hyperexcitability. Mitochondria, they are involved in ion pumping and calcium regulation. They are both required to turn cells off. So calcium regulation, mitochondria are involved in regulating calcium levels, and this is required to switch off the cells, to apply brakes on the cells. When mitochondria are not working properly, then brakes will not be applied on the cells will become hyperexcitable, and this can result in symptoms of mental disorders. If mitochondria are not working properly, then these processes of turning the cell off will take longer to perform, and the cells can become hyperexcitable. Now sometimes hyperexcitability can occur due to dysfunction in the cells that are meant to slow down other cells. Sometimes hyperexcitability can occur due to dysfunctioning of the cells that are meant to slow down other cells, like the GABA cells. Now the GABA cells, GABA is a neurotransmitter. These GABA cells, they are produced in the cortical interneurons. Now these GABA cells, they are inhibitory. They stop the hyperexcitability of the other cells. And when these GABA cells are not functioning properly, when they are slowed down, when mitochondria are not functioning properly, mitochondria in the cortical region in the brain, when they are not functioning properly, then it will lead to slowing down of these GABA cells. And these GABA cells, when they function properly, they actually inhibit the other hyperexcitable cells. And when these GABA cells are not working, then they will not perform their function of inhibiting the hyperexcitable cells, and this will lead to hyperexcitability, causing symptoms of psychosis, as we see in schizophrenia or schizoaffective disorder. So psychotic symptoms: hyperexcitability of brain cells that process perception. There are certain cells, brain cells, that are processing perception, and they are there in the cortical interneurons in the brain. And GABA is inhibitory. GABA is inhibitory in nature, and when these GABA cells are not functioning properly, then the inhibitory function will not be performed. It will fail to inhibit the hyperexcitable cells, and abnormalities found in the functioning of these GABA cells in the cortical interneurons. These may result in mental disorders like schizophrenia, Alzheimer's disease, epilepsy, and autism. Or psychotic symptoms can occur due to hyperexcitability of brain cells that create dreamlike experiences during sleep. We have just seen that during sleep, certain cells become active and we have dreams. But when these cells fail to shut off, when they fail to stop during the daytime, then we will start having dreamlike experiences even during daytime, which are known as psychotic symptoms like hallucinations or delusions, which are seen in mental disorders like schizophrenia. Now, maintenance problems can alter the structure of cells, such as problems with myelin or beta amyloid can cause hyperexcitability. We have just seen that the nerve cells, the axon in the nerve cells, they are covered by a myelin sheath. Now this is the nerve cell body, dendrites, axon, nerve endings. Now this axon is covered by myelin sheath. When this myelin is defective, when myelin is not present or it is defective, then ions can leak into the nerve cell, and this can cause the cell to fire when it should not, causing hyperexcitability. So problems with myelin and beta amyloid, it can cause hyperexcitability. We have seen that beta amyloids, it accumulates in people with Alzheimer's due to maintenance problem in the cells. So all this can cause hyperexcitability. Lack of myelin can allow ions to leak into the cell and cause it to fire when it should not. Hyperexcitability has even been measured in healthy rodents subjected to no more than chronic stress. So in animal models, we have seen that when these animals are subjected to chronic stress, then it can cause hyperexcitability of cells. So this can occur in humans as well. When there is chronic stress, then it can lead to hyperexcitability of our brain cells, causing various symptoms of mental disorders. Now, cell shrinkage and cell death. If the quantity and health of mitochondria decreases, then the cell gets stressed. When the health of mitochondria will decrease, then it will lead to stressing of the cell. And if the workforce reduces, that is, if the number of mitochondria reduces, then they cannot keep the entire cell working. Because mitochondria are the only organelles which are moving around the cell. And when mitochondria stop going to the peripheral parts of the cell, like the axon terminals or the dendrites, when they stop going to the peripheral parts of the cell, then now this is the nerve cell, this is the axon, this is the nerve ending, these are the dendrites. Now the peripheral parts of the cell are these parts. When mitochondria stop going to these parts, because they are less in number of mitochondria is less, so they stop going to the peripheral parts of the cell, and this can lead to death of these cell parts. And death of cell parts will lead to inflammation. And when inflammation occurs and the death of cell occurs, then microglia, which are the immune cells in our brain, they start working and they absorb some of these dead cell parts. So microglia, which are the immune cells of our brain, they get to work and they absorb some of the dead cell parts. And as more and more mitochondria become impaired, then more and more of the cell, it starts shrinking. So mitochondrial impairment is causing the shrinking of the cells. And as this process continues, then the cell dies. And now the brains of people with chronic mental diseases show signs of shrinkage over time. So brains shrink over time. Why? Because the number of mitochondria are less, they are not going to the peripheral parts of the cell, and because of this the cell parts are dying and the cell is shrinking and people are aging prematurely. Now different brain areas are affected in different people. Some areas of the brain, like the hippocampus, now this is our brain, this is our brain, the forebrain. Hippocampus is somewhere here. It is a part of the forebrain. It is part of the limbic system. Hippocampus is part of our limbic system, which is regulating our behavior and emotions. Now some areas like the hippocampus is somewhere here. So areas like a hippocampus are most commonly impacted by this cell shrinkage and cell death. But even in people with the same diagnosis, such as schizophrenia, there can be numerous differences in the brain regions impacted. Now two people both may be having schizophrenia, but the brain regions impacted may be different. Why? Because of mitochondrial dysfunction and dysregulation. Because these two people have come in contact with different environmental factors. One person might have had the schizophrenic symptoms might have started at age 14 only. Another person, the schizophrenic symptoms started at age 42. So this person is having more, the disease has affected this person for a longer time. So different brain regions may be impacted, different symptoms may be experienced, and this person is affected at a later age. So this person may have different symptoms because of different environmental factors, because of different pre-existing vulnerabilities. So all these factors play a role. Now we have just seen overactive brain functions, cell shrinkage and cell death, and problems in cell maintenance, which are seen in people with mental disorders. Then about underactive brain functions, when the cells are not performing their function optimally, they are performing less than the required standard, they have slowed down. So mitochondrial dysfunction and dysregulation can slow down or reduce the function of cells. Now mitochondria act as the on-off switch for the cells, turning the cell on or off. They control the calcium levels and other signals in the cell. Brain cells need energy to produce and release neurotransmitters and hormones. Now mitochondria, they are controlling the calcium levels and other signaling pathways. Brain cells are needing energy to produce and release neurotransmitters and hormones. Now, reduced cell function explains altered levels of neurotransmitters and hormones in people with mental disorders. See, if the cell is not functioning properly, mitochondria are not functioning properly, then neurotransmitter release and hormone release will be adversely affected. Now, mitochondria, they are directly involved in making certain hormones like cortisol, estrogen, testosterone, etc. Now, cortisol, we know it is a stress hormone. Estrogen is the female sex hormone, and estrogen, apart from reproductive function, it performs a large number of other functions also. When women experience a decrease in estrogen levels, then cognitive decline can also take place. Memory problems can also take place. So estrogen is playing a greater role in metabolism also, and testosterone. So all these hormones, mitochondria are directly involved in making them. Now, when brain functions are underactive, then the symptoms may wax and wane. Sometimes there may be a high peak of symptoms, then there may be a reduction. For example, in people with ADHD, that is attention deficit hyperactivity disorder, there is a reduction in activity of the norepinephrine neurons. Now, this hormone, it is related to attention. It helps people focus, plan, and to stay at work. So when there is reduction in activity of the norepinephrine neurons in a region of the brain known as the locus coeruleus. Now this locus coeruleus is a small nucleus, small group of neurons in the brain stem. So this is the brain. Then there is a brain stem. So this is the lower portion of the brain is known as brain stem. So locus coeruleus is a small group of neurons in the brain stem that produces the noradrenaline or norepinephrine hormone. And it plays a role in a lot of functions like arousal, attention, mood, memory, stress response, pain modulation, motor control, energy, homeostasis, etc. So in people with ADHD, there is a reduction in activity of norepinephrine neurons which help people focus, plan, and stay at work. So these neurons are present in locus coeruleus, and there is an abnormal activity of these neurons. There is reduction in their activity in people with ADHD. Then Alzheimer's disease. In Alzheimer's disease, reduced function of neurons involved in storage and retrieval of memories is seen. This is the cause of dementia in people with Alzheimer's disease. So reduced function of neurons involved in storage and retrieval of memories. One aspect of depression can involve reduction in activity of a brain system called the default mode network. So one aspect of depression can cause reduction in activity of a brain system known as default mode network, causing slowing or disorganization of normal brain function. So in depression, again, certain cells are underactive. Brain systems that control moods, they are underactive in mood disorders, in personality disorders, and in anxiety disorders, causing unstable moods and anger outbursts. So this is about underactive brain functions. Now we come to absence of brain functions. Certain mental disorders may involve absence of brain functions. So these are mainly the neurodevelopmental and neurodegenerative disorders. For example, autism. In autism, neurons or connections between neurons appear to be missing or at least different. So in autism, neurons, either the neurons are missing or they are different, or the connections between neurons are missing or different. So the development of neurons has not taken place properly, leading to autism. Then Alzheimer's disease. In Alzheimer's disease, again, brain shrinkage and death of neurons. So these are neurodegenerative disorders and their effects may not be reversible. Their effects may sometimes become fixed. They may become permanent. Why? Because the neurons have not developed properly, or there has been death of neurons, there has been death of brain cells. And so these become permanent or fixed. But still there are symptoms of autism and Alzheimer's which do wax and wane. And these symptoms are anxiety, psychosis, mood changes. So dementia may not wax and wane. The memory problem may become permanent. But certain symptoms can have peaks and lows, like anxiety symptoms, psychosis symptoms, mood changes, etc. So Dr. Christopher Palmer says that while diseases, psychiatric disorders like schizophrenia, schizoaffective disorder, bipolar disorder, anxiety, depression, they can be reversed. They can be reversed through metabolic interventions. But sometimes the effects of neurodegenerative disorders, they become permanent because the death of neurons, the death of cells has taken place and the effects cannot be reversed, or the development of neurons has not taken place properly. So sometimes the effects of neurodevelopmental and neurodegenerative disorders cannot be reversed. But the effects of psychiatric disorders, there is hope that these effects can be reduced or reversed with metabolic interventions. When mitochondrial dysfunction and dysregulated brain doesn't develop normally. So when the mitochondria are dysfunctional or they are dysregulated, then the brain will not develop normally. So again, these neurodevelopmental and neurodegenerative disorders, they are due to dysfunctioning of mitochondria or dysregulation of mitochondria. That is, the environmental factors are adversely affecting the function of mitochondria, that is dysregulation, or the mitochondria are themselves dysfunctional due to some mitochondrial DNA mutation. So all these things happen in case of neurodevelopmental and neurodegenerative disorders also, and the brain doesn't develop normally. Now we come to the numerous factors that are affecting the mitochondrial function. What are these numerous factors that are affecting the mitochondrial function? These factors include, first, genetics and epigenetics. Genetics means genes, and that is the hereditary characteristics, and epigenetics means turning of the genes on or off, the mechanism by which genes are turned on and off. So it is mainly the epigenetic mechanisms which are influencing mitochondrial function. Then other factors are the chemical imbalances, the neurotransmitters and hormones and medications. So neurotransmitters like serotonin, dopamine, glutamate, GABA, norepinephrine, all these neurotransmitters, they are affecting mitochondrial function. And medication, the psychiatric medication as well as certain metabolic medications, they are also affecting the mitochondrial function. Then hormones and metabolic regulators. Hormones are also affecting the mitochondrial function. Hormones like thyroid hormone, then estrogen hormone, they are also affecting the insulin. Insulin is a major hormone which is affecting the mitochondria. Then inflammation. Inflammation is directly affecting the mitochondria. And in most of these factors, there is a feedback mechanism. Like inflammation is causing mitochondrial dysfunction. And sometimes mitochondrial dysfunction will lead to inflammation. Mitochondria are producing neurotransmitters, and neurotransmitters are in turn affecting the mitochondria. Mitochondria are playing a role in production of hormones, and hormones are in turn affecting the mitochondria. So there is a feedback mechanism everywhere. Then sleep, light and circadian rhythms. So the time that you wake up, the time that you sleep, the exposure to light, all of this affects mitochondrial function. Then food, the food that we eat, fasting and gut microbiome, they also affect mitochondrial function. So feed the brain what it requires. When you feed the brain what it requires, then it can provide the key to cure for a large number of mental as well as metabolic disorders. Then about drugs and alcohol. Drugs and alcohols also affect the mitochondrial function. They contribute to dysfunctioning of mitochondria. Then physical activity, that is exercise. Exercise also leads to formation of new neural connections, new neurons in the hippocampus in our brain. Then love, adversity and purpose in life. So psychological and social factors are also playing a major role in the mitochondrial functioning. Now different factors are affecting different areas of the brain. So depending on the mix of risk factors or causes that people have, their brains will be affected differently. So different people will be have been exposed to different environmental factors. So depending on the different inputs, different people with the same diagnosis may have different symptoms. Now timing and development also matter. Now people who are affected at age 14 will have brain differences from the people not affected till the age of 39. So suppose one person is having schizophrenia at age 14 and another is developing the disorder at the age of 42 or 39, then there will be brain differences in these two persons. So there will be differences in the symptoms experienced by both these persons. So these are all the risk factors that are causing mitochondrial dysfunction. Now we come to major depression. Now several lines of evidence have found that people with depression have lower levels of ATP, that is they have lower levels of energy. There is underactive brain cell function. Underactive cell function not only in the brain cell but also in the muscle cell and circulating immune cells. So there is low energy production, there is underactivity of cells in case of depression. Now autopsy studies looking at the brain tissue of people with chronic depression found specific abnormalities in metabolic proteins, clearly implicating mitochondrial dysfunction. So mitochondria were dysfunctioning, because of which there were abnormalities in metabolic proteins, and these abnormalities in metabolic proteins were seen in people with depression. And levels of oxidative stress, that is high number of reactive oxygen species, were also seen in people with chronic depression. So these are clearly connected to mitochondria. So mitochondrial dysfunction is causing symptoms of depression, is becoming a common pathway to depression and to other metabolic and mental disorders as well. So blood biomarkers of depression, like the doctors took blood samples of people with depression and compared them to healthy controls. The researchers took blood samples of people with depression, compared them to healthy controls, and the biomarkers related primarily to amino acid and lipid metabolism. So the biomarkers that were related to amino acids, we know they are the basic constituents of proteins, and lipids include fat, oil, and cholesterol. So biomarkers that were related to amino acid and lipid metabolism, both of which related to mitochondrial function, these biomarkers were showing differences. Now acetyl L-carnitine, it is a molecule which is produced within mitochondria and it is important for energy production within the mitochondria. And this molecule is critical to the functioning of hippocampus. And this hippocampus, we have already seen, it is a brain region. Hippocampus is a part of the limbic system, and it is involved in the regulation of our limbic system is involved in the regulation of our emotions and behavior. And hippocampus is also involved in the consolidation of our memories, converting of short-term memory into long-term memory. So this molecule, acetyl L-carnitine, is critical to the function of hippocampus. And this hippocampus is a brain region which is often implicated in depression. So depression is affecting the hippocampus. Study found that depressed people on an average had lower levels of this molecule, acetyl L-carnitine. And this molecule is produced within mitochondria. Again, we can see a clear connection between mitochondria and depression. Another study says that acetyl L-carnitine levels improved with effective antidepressants treatment. So when antidepressants were given, then the ALC levels improved. Now antidepressants, they were working by affecting the functioning of mitochondria, and when antidepressants were given, then the ALC levels improved and there was improvement in depression symptoms. These levels could help predict who would experience full remission of symptoms. Now see, there is an enzyme called as monoamine oxidase. This enzyme, monoamine oxidase, it is involved in the degradation of certain hormones like dopamine. serotonin, norepinephrine, etc. So it is involved in the degradation of certain hormones, and we know that these hormones they influence the symptoms of depression. And monoamine oxidase is involved in the degradation of these hormones. So antidepressants, they act as monoamine oxidase inhibitors. Antidepressants, what are they doing? They are suppressing the activity of this enzyme monoamine oxidase. So when the activity of this enzyme monoamine oxidase, which is present in the mitochondria, this enzyme is present in the mitochondria, when its activity is inhibited, then it will mean that this enzyme is now not degrading the hormones like dopamine, serotonin, etc., leading to increase in the levels of hormones like serotonin, and increase in hormones like serotonin will reduce the symptoms of depression. So ALC levels improved with effective antidepressant treatments, and these levels could help predict who would experience full remission. So in short, acetyl L-carnitine is a molecule produced within the mitochondria, and it is important for energy production, and it is critical to the functioning of hippocampus, which is a brain region implicated in depression. And depressed people had lower levels of this ALC molecule, and when antidepressants were given, then ALC levels improved, and this led to reduction in depression symptoms. So ALC levels improved with effective antidepressant treatments, and these levels could help predict who would experience full remission from the symptoms of depression. Now we come to a study in rats. Difference in mitochondrial function and or how the cells developed in specific part of their brain, the nucleus accumbens. So in this part of brain called the nucleus accumbens, which is a small structure in the brain that plays a role in reward, motivation and decision making and more. So this region of the brain, it plays a role in reward, in motivation and decision making and even other functions. So there was a study conducted in rats, and the study found a difference in the mitochondrial function and how the cells developed in the nucleus accumbens in the brains. The anxious and depressed rats had fewer mitochondria per cell in the nucleus accumbens. So when the rats who were anxious or depressed, they had less number of mitochondria. And also there was a difference in the way their mitochondria used oxygen to turn energy into ATP, and how the mitochondria interacted with another organelle called the endoplasmic reticulum. And the neurons themselves, they also look different. So there were fewer mitochondria in the anxious or depressed rats in this region of the brain. Now following the trail further, the researchers found that mitochondria from these rats had lower levels of a protein known as mitofusin 2. This protein is there on the mitochondrial membranes, and this protein, that is mitofusin 2, it is important to the ability of mitochondria to fuse with each other. So this protein is facilitating the fusion of mitochondria. And the rats who were anxious or depressed, they were having lower levels of this protein mitofusin 2. And so when the levels of mitofusin 2 were lower, then mitochondria were unable to fuse with each other, and they were unable to interact with the endoplasmic reticulum, and this caused symptoms of anxiety and depression. So there was lower levels of this protein which was adversely affecting the functioning of mitochondria, and which was causing these anxiety depression symptoms. So what the researchers did is that they injected the mitochondria with a viral vector that increased the levels of this mitofusin 2. And when the levels of this mitofusin 2 were increased, then mitochondria, they started functioning normally, and the symptoms of anxiety and depression, they stopped. So we can see that mitochondria are influencing, they are directly playing a role in depression and anxiety. Mitochondrial dysfunctioning and dysregulation is leading to anxiety and depression. So in anxiety depression, what happens is number of mitochondria reduce, and there is a protein called mitofusin 2, which is there in the mitochondrial membrane, which impacts the ability of mitochondria to fuse with each other. And the levels of this protein, mitofusin 2, these are less in case of anxiety and depression, as seen in the rats. Now, depression symptoms include changes in sleep, energy, motivation, concentration, reduced functioning of the brain cells, leaden paralysis. Then there is also a symptom known as catatonia, which is seen in many mental disorders. It is an extreme version of metabolic failure because people can appear paralyzed. People can appear paralyzed from their illness, and they can have severe difficulty moving or speaking. So people with mental disorders, some of them experience this extreme symptom called catatonia, in which they appear to be paralyzed. They are not able to move around. They are not able to speak. And mitochondrial dysfunction is directly affecting the mental disorders, the onset of mental disorders, or the exacerbation of mental disorders. Now about bipolar disorder. Again, even in bipolar disorder, direct evidence of mitochondrial abnormalities were found in bipolar disorders and even in schizophrenia. It dates back to 1956 when researchers noted abnormalities in lactate metabolism in patients. So, researchers found abnormalities in the lactate metabolism in the patients. So, what happened in case of bipolar in the manic states was, in bipolar disorder there are depressive states and manic states. So, person will have mood swings, sometimes depression, sometimes hypomania, that the person becomes highly excited, and sometimes the person becomes extremely depressed. So what happened in manic state was that there was increased glucose and lactate utilization in the brain. So the utilization of glucose and lactate in the brain was increased during the manic states, which suggested increased mitochondrial energy production. So increased energy production during manic states. Two neurotransmitters, glutamate and dopamine, were found to be elevated in the manic states. So two neurotransmitters, glutamate and dopamine, they were found to be elevated in the manic states, which suggested increased activity of neurons. Because these neurotransmitters, they were elevated, it suggested increased activity of neurons, increased energy production, leading to manic states. And manic states appear to be one of the few unique situations in which mitochondria, at least in some brain cells, are producing more energy than normal. So in manic states, in some brain cells, the mitochondria, they are producing more energy than normal. And this is also a sign of mitochondrial dysfunction. Because when they should not be producing excess energy, they are producing. So in some brain regions, they are producing more energy than normal. Mitochondria are becoming hyperactive, and they are making the cells fire. They are making the cells go when they shouldn't. And many parts of the brain, they appear to be overactive. Now, bipolar patients were found to have higher than normal calcium levels. So in bipolar patients, the levels of calcium were higher than normal, especially when they were manic. So when the patients were in a manic state, they were extremely excited. So at that time, higher than normal calcium levels were found, and this is a sign of hyperexcitability. In people with bipolar disorder, once the manic state resolves, now in bipolar disorder, there is a manic state, and then there will be a depressive state, extreme mania, extreme depression. So once the manic episode is gone, then the mitochondrial dysfunction continues. And in the manic episode, there is too much of energy. Mitochondria are producing too much of energy. Then when the depressive state comes, the mitochondria are producing too little energy. So this is a sign of mitochondrial dysfunction and too little energy production during the depressive state. Now, one group of researchers identified a mitochondrial biomarker in blood cells showing a reduction in number of mitochondria in both manic and depressed states. So in a blood biomarker, when they saw the blood cell, they identified a biomarker which showed that mitochondrial activity in the number of mitochondria was reduced in both the manic and depressed states. And when the people were feeling well, the number of mitochondria was normal. But when they were in manic or depressed state, number of mitochondria reduced. So in case of mental disorders, what happens is that the cells are firing when they don't need to. The cells are either overactive or they are underactive. Now Dr. Palmer tries to explain all this with the example of three cars. Three cars. Now we'll just discuss that example. What Dr. Palmer says is, suppose there are three cars A, B and C, and these three cars are of the same make and model. That means when you take this analogy to people, three people and they have the same genes, they are of the same. Now these three cars, they are of the same make and model. Now car A, it is there in California. In California the weather is good. The car A, the driver does not take, and the car A does not drive too often on the roads. It comes out very less. It is mostly there, mostly standing in the garage. So it is getting used very less and the weather is fine. So there is no maintenance problem with car A. And the driver takes the car out only, suppose, for one day or 2 days during the week. So there is no problem with car A, and car A will naturally have a longer lifespan. Now car B. Now car B is there in New Hampshire. Now New Hampshire in USA is a hilly area. So it is there in a hilly area and there are frequent blizzards. The winters are very harsh. They are fierce winters. So car B, when it is, the driver is also taking the car out very often during the weekdays and it is getting used too much. And now when it is driving on the rough terrain, then it needs to switch on its four-wheel drive. It needs to switch on its wipers, windshield wipers when it is raining or when there is a storm. And it needs to switch on its blinkers. It needs to switch on its lights because it is frequently being driven during the night time. So there are adverse, the environmental factors are adverse. So car B is subjected to a lot of stress naturally. So it is turning on only all these adaptive mechanisms like wipers, lights, four-wheel drive. And so car B, since it is living in adverse conditions, it is natural that car B may have a shorter lifespan because it is getting used too much. Now we take the case of car C. Now what happens in case of car C is that car C is living at a place where the weather is not that harsh, not as harsh as New Hampshire, and the winters are also not very fierce. But in case of car C, what happens is that car C is turning on its wipers even in sunny weather. So even in sunny weather, it is turning on its wipers, and this is causing the windshield to get scratched. So it is performing a function when the function is not needed in reality. Now car C is switching on its lights in daytime and it is not switching on its lights during night. Car C is not switching on its lights during the night. So it is when the function is needed, it needs to switch on its lights, it is not doing so. So this means underactive function. It is switching on its wipers even during sunny weather. So this is overactive or hyperexcitability. So it is doing things when the things are not needed. And car C is driving at a very slow speed, only at 25 km per hour, when the other cars are going at 60 km per hour. So car C clearly shows a mental disorder because car C is doing things which are not required, doing things at the wrong time, in the wrong way. So car C is having a mental disorder. Car B is not having a mental disorder. It is naturally adapting to adversity. Now car B is turning on its wipers. It is turning on its four-wheel drive because it needs to do so. So if car B is having stress, it is having symptoms like anxiety, depression, it is quite normal because the environmental factors are such that it needs to have these adaptive mechanisms. But when car C is doing things at the wrong time, it shows a mental disorder. So car C may meet with an accident also. and car C might end up dying an early death. So car C is showing, is an example, is symbolizing people with mental disorders. So the cells are doing its task when they should not be doing it, or the cells are not doing the task when they should be doing it. This indicates a disorder. Now, Dr. Chris Palmer further says that the problem may not be with the cars themselves, the problem may be with the drivers of the cars. Now, the driver of the car C is not in the proper mental state. So, the drivers of our cells, they are the mitochondria. So, just as the drivers of these cars are playing an important role in the functioning of these cars, in maintenance of these cars, so the drivers of our cells, mitochondria, are playing an important role in the maintenance and health of our cells. Now we were on bipolar disorder. Bipolar patients found to have higher than normal calcium levels, especially when they are manic. Now, one group of researchers identified a mitochondrial biomarker in blood cells showing a reduction in number of mitochondria in both manic as well as depressed states that normalized when people were feeling well. This suggests that something might be disrupting either mitochondrial biogenesis or mitophagy. So something was disrupting either the making of new mitochondria or the repairing of mitochondria throughout the body during disease, not just the brain. So the entire metabolism was affected, the entire body was affected, not just the brain. Now we come to another disorder, that is post-traumatic stress disorder. This is also an example of hyperexcitability, hyperexcitable trauma response system. Now, when we experience trauma, then we have a normal response to life-threatening events. What do we have? Stress response to these life-threatening events. Now, when this stress response is turning on when it should not. Now, during times of trauma, it is natural to be afraid. It is natural to have anxiety. But when we are having this fear and anxiety, even when trauma is not present, then it shows hyperexcitability. So the cells that are influencing our fear response or anxiety response, they become hyperexcitable. So they are turning on when they should not be turning on. So this is a sign of post-traumatic stress disorder. For some people, it appears to develop at a lower threshold for firing. So suppose now it is natural to feel fear when there is a lion in front of you. But when you are scared, when even at a lower threshold we start getting scared. When we are scared without any reason. So that shows a disorder. Now people have clearly identified triggers in case of this disorder. Now two areas of the brain are affected in this post-traumatic stress disorder. The first area is the amygdala. Amygdala is again a part of our limbic system, which regulates our behavior and emotions. And the other area is the medial prefrontal cortex. Now, amygdala in our brain. Amygdala is located close to the hippocampus in the brain. Amygdala sets off a fear response and has been found to be hyperexcitable in PTSD. So the fear response is hyperexcitable. Then medial prefrontal cortex is an area of the brain that inhibits the amygdala. So when the amygdala is functioning and when it needs to stop, then the medial prefrontal cortex, it directs it to stop. But when this medial prefrontal cortex does not perform its function, then it may lead to hyperexcitability. Now medial prefrontal cortex can stop panic reactions once someone realizes that there is no need to panic. This medial prefrontal cortex of the brain, it is underactive in case of post-traumatic stress disorder. It is underactive in case of PTSD. Now studies have shown mitochondrial dysfunction in people with PTSD, including autopsy studies showing abnormalities in mitochondrial gene expression, decrease in the total number of mitochondria, abnormalities in gene expression, that is epigenetics. Now we know that mitochondria influence the gene expression. They influence which genes are turning on or off. They are involved in the calcium regulation, which is important for the off switch in the cells. And even mitochondria, through the reactive oxygen species, they are controlling gene expression. Then there was a decrease in the total number of mitochondria in people with PTSD. There were increased levels of oxidative stress or reactive oxygen species in people with PTSD. Also, there were reduced levels of ATP, that is, reduced energy production in people with PTSD. Now we come to a unifying example, that is delirium. This shows how metabolic disorders are and mental, how there is a bidirectional relationship between metabolic and mental disorders. Now, delirium, what is delirium? It is an acute mental disturbance. Delirium is an acute mental disturbance. Serious disturbances in mental abilities that results in confused thinking and reduced awareness of surroundings. Now, people having certain metabolic disorders like heart attack or cancer, they may experience an acute mental disturbance which is known as delirium. Now, this shows a clear connection between the mental and the metabolic. What causes delirium? No one knows what causes delirium. No one knows for sure how it all works, but we know that when people are seriously ill, then delirium can happen. Almost all medical disorders, they cause delirium. Almost all medical disorders cause delirium. For example, infections, cancer, autoimmune diseases, heart attack, strokes, etc. They all cause delirium. And in the elderly, in the old people, urinary tract infection is a common cause of delirium. And when this urinary tract infection occurs, then the brain is affected first because it is the weakest link. Brain is most sensitive, it is the weakest link. It requires maximum energy, more energy compared to other body parts. So it shows the first signs of the problem. Now, symptoms of delirium usually go away once the underlying medical condition resolves. Suppose delirium is due to heart condition. And when the heart condition resolves, then the delirium will go away. So this shows a clear connection between metabolic abnormalities and mental abnormalities. High rates of dementia, that is memory loss, and cognitive impairment have been consistently documented at 3, 12, and 18 months after hospital discharge in people with delirium. So when people suffering from delirium, 3 months or 12 months or 18 months after their hospital discharge, signs like dementia, high rates of dementia, cognitive impairment have been seen. High rates have been seen compared to people with the same illness who don't have delirium. So some people think that delirium is just related to the underlying cause. When the underlying cause, that is the heart condition or whatever the underlying cause is, like cancer or autoimmune disease, once that goes away, then the delirium will go away. But we need to take delirium seriously as well, because delirium, if not treated properly, it may lead to dementia and cognitive impairment. Then delirium in older people results in an 8-fold increase of subsequent dementia. So delirium in older people results in 8 times more risk of subsequent dementia, that is memory loss. Now, hyperexcitable brain cells. Now, delirium can cause hyperexcitability in brain cells, and it can cause seizures, that is abnormal electrical activity in the brain. And seizures are the most extreme consequence of these hyperexcitable brain cells caused due to delirium. Now, in a study of people with delirium, 84% had abnormal electroencephalograms and 15% showed clear seizure activity. During a hospital admission, people with delirium are twice as likely to die as those without delirium. So people with delirium, they are more vulnerable, they are twice as likely to die. After discharge from hospital, the 1-year mortality rate of people with delirium are 35 to 40% higher than those without delirium. So after discharge from hospital, the 1-year mortality rate for people with delirium are 35 to 40% higher than those without delirium. Now, delirium tells us that there is serious mitochondrial dysfunction in the brain. So mitochondria, they are dysfunctioning. And this is causing this acute mental disturbance called as delirium. Chronic mental illnesses give us a window into a person's metabolic health. Now, when a person is suffering from a chronic mental illness, it shows the poor metabolic health. They tell us that the brain is not working due to metabolic or mitochondrial dysfunction. And if we ignore this problem, if we ignore this mitochondrial dysfunction, then sometimes it can correct itself if the damage is not too much. But if it continues and we still turn a blind eye, then the symptoms and other illnesses can follow. So if we don't pay attention to it, then other illnesses can occur, because all illnesses, mental as well as metabolic illnesses, they are all connected, and they are occurring through a common pathway called mitochondria, according to Dr. Christopher Palmer. Now, the medical field says that the process of dying involves seizures, coma, and death. It is commonly said that the process of dying involves seizures. First seizure, that is abnormal electrical activity in the brain, then coma, and then death. But it should be corrected. The process of dying first includes delirium. First, there is an acute mental disturbance, confusion, then seizures occur, followed by coma and death. This is according to the theory of brain energy. The theory of brain energy suggests that all mental disorders have a common pathway of mitochondria. People with exactly the same diagnosis can have different symptoms. So people with exactly the same diagnosis, they can have different symptoms. This is because of why people with same diagnosis can have different symptoms, because of different pre-existing vulnerabilities. That is comorbidities. The persons might have other pre-existing vulnerabilities which affect the symptoms, and also because of different inputs to different cells, different environmental factors affecting different brain regions, and this can lead to people with the same diagnosis having different symptoms. Mitochondrial dysfunction and dysregulation provides an explanation for an unlimited number of symptoms in different people. Now, depending upon which brain cells and brain networks are involved, so depending on which brain cells are involved, which brain networks are involved, and which factors are affecting the functions of mitochondria, people will have different symptoms. So depending on which brain cells are involved, which brain networks are involved, which factors are affecting the functioning of mitochondria. So different environmental factors may be affecting the functioning of mitochondria. They may include hormones, neurotransmitters, neuropeptides, alcohol, drugs, psychiatric medications, etc. And these factors may affect different brain cells, different brain networks. So people will have different symptoms. We could call mental disorders metabolic dysfunction. For example, schizophrenia can be called a metabolic brain dysfunction with psychotic, depressive, and cognitive symptoms. Likewise, depression or anxiety can be called a metabolic brain dysfunction with depressive symptoms. Or bipolar disorder can be called a metabolic brain dysfunction with manic and depressive symptoms. So, what Dr. Palmer is suggesting is that all mental disorders have a metabolic angle to them. The cause is in the metabolism, and correcting the metabolism can help in correcting or reversing the mental disorders. Now, the treatments that we are giving for mental disorders, they are symptomatic treatments. They are not addressing the root cause, and the root cause is metabolic dysfunction through a common pathway, which is the mitochondria. Now, why different people have different symptoms and disorders, and why connections persist across them? If all mental disorders are due to mitochondrial dysfunction, if all mental disorders are due to dysfunctioning mitochondria or mitochondrial dysregulation, that is environmental factors adversely affecting functioning of mitochondria, then why is there so much variability in symptoms? Why are the symptoms so much variable? One person is having depression symptoms, one person is having symptoms of bipolar, another person is having symptoms of schizophrenia or schizoaffective disorder, one person is having depressive and manic symptoms. So all these depressive manic symptoms in case of bipolar. So why is there so much of variability in symptoms? How do malfunctioning? Now we are saying that mitochondrial dysfunction or dysregulation is the root cause of these mental and metabolic disorders, then the symptoms should also be same, but the symptoms are very variable. Why? So they are variable because of different pre-existing vulnerabilities, because of different environmental factors affecting the mitochondria and affecting different regions of the brain. How do malfunctioning mitochondria and metabolic toll cause one person to end up with depression and another to end up with obsessive compulsive disorder? Now there are two answers. One is difference in pre-existing vulnerabilities, nature and nurture. Another is difference in inputs. Now cells and mitochondria within them, they are affected by many inputs, ones that affect different parts of the body. So different inputs can affect different parts of the body and brain at different times. Now one person might have a disorder schizophrenia at 14, another may have it at 42. And these environmental factors, one person may have adverse effect in one part of the brain, another person may have adverse effect in another part of the brain. So one person may be affected by one environmental factor, another may be affected by another factor. So these are responsible for the variability in symptoms. Many of these factors affect only some cells and not others. When faced with different situations or tasks, different regions of the brain or body part need different amounts of energy. So when faced with different situations, so when the environmental factors are different, then the brain regions will need different amount of energy to face these environmental situations, to adapt to these environmental factors, and this also is a cause for the variability in symptoms. Some of these contributing causes will impact some brain regions and not others, and this is responsible for the difference in symptoms. Now about treatments and success stories. Now what are the different treatments that we can use for treating of mental disorders? So there are three categories of treatments. First is treatments that remove or reduce things that are dysregulating mitochondria. So certain environmental factors are dysregulating mitochondria. Suppose, for example, alcohol use or drug use. They are dysregulating the mitochondria. Certain poor diet may be dysregulating the mitochondria. Sleep disturbances may be adversely affecting mitochondria. Some medications, psychiatric medications or other metabolic medications, they may be dysregulating mitochondria. So if you remove these factors, then, or certain social stressors, suppose domestic abuse, or psychological or social stressors like some domestic abuse or some other factors, they may also lead to mitochondrial dysfunction. So if you remove these factors or you reduce the impact of these factors, then they may form a part of the treatment. So first category is this. Second kind of treatment is treatments that correct for metabolic imbalances. Now the person has got imbalance in the metabolism, imbalanced level of hormones or imbalanced level of neurotransmitters. So correcting these imbalances by giving the relevant hormone or neurotransmitter, that can also correct the problem. That is the second type of treatment. And the third type of treatment is treatments that improve the metabolism, that improve the mitochondrial functioning. Now what are these treatments that improve metabolism? These treatments can be categorized into three categories. First is treatments that these treatments, they are going to improve metabolism. How? They are going to improve metabolism in three ways. Now what are these three ways? One is increasing the number of mitochondria by producing new mitochondria, mitochondrial biogenesis. Second is repairing the defective mitochondria, getting rid of old defective mitochondria, replacing them with new healthy mitochondria. And third is repairing the cells. Third is repairing the cells, that is autophagy. So repairing mitochondria is mitophagy, and repairing cells, repairing the structural damage that has occurred to cells because of long-standing metabolic problems. And this can be essential to long-term healing. So repairing the functioning of cells, repairing the old and damaged cell parts, recycling them, making new cell parts, this is autophagy. So these treatments which improve metabolism, they work by three ways: increasing the number of mitochondria, repairing the mitochondria, or repairing the cells. recycling the defective cell parts. And these treatments can include dietary changes in the diet. For example, in the later slides, we will be studying about a kind of diet called the ketogenic diet. Now, this ketogenic diet, it is improving the metabolism. It is promoting autophagy and mitophagy. So this is a fasting mimicking diet that is promoting mitochondrial health. And this diet is a metabolic intervention for promoting better metabolic health in people with mental disorders. And this diet has got less than 20 grams of protein, less than 20 gram of carbohydrate, and moderate amount of protein and fat. It has got less than 20 gram carbohydrate. And what does that mean? Very few calories are obtained from carbohydrate, and protein intake can be up to 70 grams per day. 20 to 35% of calories are coming from proteins and some fat also. So when the body is not getting carbohydrates, then the body will try to make energy from fat. Liver will try to change fat into energy, will try to change fat into ketones. And these ketones, they are beneficial for the brain mechanism. We will study in the later slides that these ketones act as a rescue energy source for insulin resistant brain cells. So ketogenic diet has been found in some patients to be a beneficial intervention for providing remission from mental disorders. We will study all this in detail in the later slides. So we have studied about the brain energy imbalance contributing to mental disorders. And this was the part 2 of the summary of the book Brain Energy by Dr. Christopher Palmer. In the part 3, we are going to study about the different contributing causes to mental and metabolic disorders and about certain beneficial interventions that can promote better mental health. Now, one more thing is that the author says that intermittent fasting, it is also a way of promoting good metabolic health, because intermittent fasting also promotes autophagy and mitophagy. We will study all these in detail in the subsequent slides in the part 3 of the summary of the book Brain Energy. Thank you.